Hi, my name is George. I am a postdoc at the Dementia Research Centre here at University College London, and I'm very pleased to be giving the final lecture for this online SPM course. And I'm going to be talking to you about dynamic causal modelling for resting state functional MRI data. This is going to follow on hopefully nicely from the talks you've already had about Bayesian inference and Bayesian statistics, and also quite closely is going to follow on from the lecture you've just had from Peter Zeidman in his introduction to DCM and the principles of DCM. And I'm going to be speaking a bit more specifically about DCM for resting state fMRI. So, some learning objectives, what we should hopefully know by the end of this lecture. Number one is when and why you should use a resting state DCM. Number two is how it differs from a conventional DCM, or you might think of that as a task-based DCM. Related to point two is what is a spectral DCM, or dynamic causal modeling. And then finally, once you have your subject level DCM specified, how do you then take these to the group level and use them to test specific hypotheses that you have? This is going to be more extensively covered in the special interest practical session with me, if you're attending that. But I'll also go over it at the end of this lecture, just to outline how it looks. So, point one, why and when you should use a resting state DCM. So why use resting state dynamic causal modeling? Well, first of all, you have or are planning to collect some resting state fMRI data. You have or are planning to collect some anatomical data. And most importantly, you have a question that you want to answer. DCM is not an exploratory methodology. It's designed specifically to test hypotheses you have about your data and to ask specific questions. So point three here is really the most important one. Obviously you need the data, but you really do need to have a question you want to answer if you want to do some DCM. You'll see that I've got a little asterisk here on point one, and that's kind of related to this final point, which is if you do have this specific question, you haven't yet collected your functional MRI data, you might ask whether a task should be better. You are relinquishing quite a lot of experimental control by going to resting state. And it might be the case that if you can design a task which more specifically probes the question that you're interested in, you might be able to employ DCM more effectively. However, it might be the case that your hypothesis is one that relates to resting state or doesn't require people to do a task. It might be the case that you are working with a clinical population or group of people who find it hard to perform tasks in an MRI scanner. Or it might be the case that you already have some resting state data collected and want to try and do some dynamic causal modeling on it. And yeah, that moves us on to what we need to run a DCM. So what do you need? This is going to look familiar. You need some resting state fMRI data. You need some anatomical data. And this anatomical data is going to be used, like with all kind of fMRI analyses, you need this alongside your functional data to co-register between the functional and the anatomical data, to bring everyone into the same anatomical space and to help with definition of regions of interest and things like that. So that's that. And then finally, you need a question that you want to answer. This is where things differ the most from the task-based DCM. So this question that you want to answer is not going to be driven by task activation, but is likely to be related to group differences rather than differences in task performance. For this question, you need some predefined regions of interest, regions where you think there's going to be some group differences, and some hypotheses about how you think these regions are communicating with one another. So I'll talk a bit about defining hypotheses now. Just to hammer this point home, DCM is not an exploratory methodology. You shouldn't go in with a group of regions without a specific idea of what question you're trying to answer, and just go in like that and run a DCM and find some differences, because it will give you an answer. It will be optimized to try and give you an answer that makes sense. But if you don't structure your question well, then the answer you get out is not going to be particularly informative. So how do you define your hypothesis then? Well, you don't have a task, like I said, so hypotheses are likely to be related to group differences. And you're basically going to be thinking about which regions of interest do you think are important in your groups. This might be something like one group who have been given a drug versus a placebo, or it might be differences between a disease group and a control group. And then you'll have some hypotheses about how you think directional effective connectivity between these regions is going to change as a result of being given this drug or being part of this disease group, as opposed to a control group. This region selection is obviously quite important, but how do you choose your regions? Like I said, this is not going to be driven by task activation, so you need to be a bit more a priori about what kind of regions you're going to be choosing. Some other options for how you could choose your regions include from the literature. If there are some reviews which give you MNI space coordinates, for example, you could use them to define your regions. You could base this off a structural segmentation of your anatomical data. If you just have one region that you're interested in and want to see how it's connected to other regions, you could do some seed-based functional connectivity at first, and then use that mask to define other regions and then test the effective connectivity between them. Or you could do some ICA type analyses to pull out the canonical resting state networks and compare the effective connectivity within them. I would be a bit cautious about this last one because, as we've discussed, all of this really needs to be driven by your hypothesis. If you have a specific hypothesis about, for example, the default mode network, then fine, you can run your ICA and put in the regions you get out and ask some questions about that. But I would caution against just running an ICA analysis, putting in the DMN regions, for example, and running a DCM based on that, because you will get an answer, but it might not be particularly informative if you haven't decided on what question you're actually trying to ask. So that's hopefully learning point one sort of done for now. I'm now going to talk about how a resting state DCM differs from a task DCM. Here I have a cartoon example of how a task-based DCM might normally be defined. So you will have defined some regions of interest within the brain. And between these regions of interest, you will have defined some extrinsic connections that define how activity in one region is causally influencing activity in another region. You will also have some within region or intrinsic connections that will act to, over time, quell the activity of a region which is active to avoid kind of runaway positive feedback. You'll have some experimental driving inputs to particular regions, so inputs that might act to increase or suppress the activity in a particular region. And you'll finally have some modulatory inputs, so how does your task modulate the connections that you have between or within regions? Does it make them stronger or weaker? But as you can probably see, we actually have more information here than we need because there are no external inputs in resting state fMRI, or at least no task-based external inputs. And that means that we don't need these modulatory or driving connections in this cartoon example here. So I'm now showing you the same thing that I showed you on the previous slide, but in the form of equations. And specifically, this is what's known as a Taylor expansion of the neural model. I'm not going to go into how this is derived here, but if you are interested, I can show you, or I can point you rather towards Peter's paper, which has a derivation of it. But basically what this is showing you is the neural model of the DCM. And it's showing you that the changes in neuronal states over time are dependent upon the connectivity between regions, which is this matrix J, and some driving experimental input described by C and the vector of experimental inputs U. This J matrix is dependent upon the connectivity between regions, as you might expect, which is this A matrix here. And then also is dependent upon the modulatory inputs on those connections, described by this B matrix, and again, the vector of inputs U. But because we have resting state rather than task-based data, we don't have any driving experimental input. We don't have any modulatory input. And in fact, we don't have any inputs at all. And all we need to define is this A matrix, which specifically comprises the intrinsic self-inhibitory connections constrained to be negative, and the extrinsic connections between regions. And these are, in fact, the only things that you need to define or specify when you're trying to build your subject level DCM for resting state fMRI. You don't need to define the B matrix or the C matrix or U as you would for the task-based DCM. We have a problem here, though, which is that we can't just have the changes in neuronal states over time be dependent upon the connectivity between regions, because nothing would happen. There is nothing driving this system at the moment, and that's not going to be useful if we want to try and simulate some functional MRI data, which is what we do in DCM. So what's driving the system? In order to understand what's driving this resting state system, we can have another look at the neural model of the DCM. So this is the neural model again, this time in continuous form rather than in the form of the Taylor expansion, which I showed you on the last slide. And just to recap what this is showing, the change in neuronal states over time, Z dot, can be defined as a function of these hidden neuronal states, which is what we're trying to get at with DCM, and some connectivity parameters, which are going to be within some neural parameters. And these connectivity parameters that we need to specify are going to end up being the extrinsic connections between regions and the intrinsic self-connections. But what's driving the system? Well, in the absence of any external experimental input, as is the case with resting state fMRI, we assume that the system is going to be driven by endogenous fluctuations in neuronal activity of the type that you might expect in any kind of biological system. So yeah, it's going to be these random changes in neural activity rather than experimental driving inputs, which is going to give this system some kind of impetus. But there's a problem here as well, which is that these fluctuations are time varying and we don't know what they are. So whereas with task-based DCM, you could define your inputs driving the system, here these endogenous neuronal fluctuations are another thing that you have to estimate. And estimating them in this form, in their time-varying form, is incredibly computationally complicated and difficult. And this is where spectral DCM comes in. So this moves us on to our third learning objective, and that is, what is spectral DCM? Well, spectral DCM is just a way that you can do resting state dynamic causal modeling. And it's a way that actually is going to significantly simplify this computation that we need to do because of the fact that we need to model the driving input in our system, which is going to be these endogenous neuronal fluctuations. Spectral DCM is by far the most common way of doing resting state DCM, and I am going to talk a bit about that now. So when we're doing our model inversion, what we're trying to do is estimate parameters that are going to best simulate the fMRI data that we have, essentially. But as I've said, if we try and do this in the time domain for resting state fMRI, it's going to be very complicated because of these endogenous neuronal fluctuations. So how can we simplify this inversion? How can we simplify the computation? Well, it turns out that one thing we can do is, rather than modeling time series as we do with conventional task-based DCM, we can instead model what's known as cross-spectral densities. So on the left-hand side here, you can see some toy cartoon time series from a few brain regions. And these are the things which you're normally going to be simulating with your forward DCM model when you're doing a conventional task-based DCM. In spectral DCM, what we're instead going to be trying to simulate is this cross-spectral density here. And these two are related to one another through Fourier transform and inverse Fourier transform. I put an asterisk here because it's not actually a Fourier transform of the time series directly, which gives you the cross-spectral density, but rather a Fourier transform of the cross-correlation function. So what is the cross-correlation function? Well, the cross-correlation function is a function that describes the correlation in activity between any two brain regions, I and J, at different temporal lags. So you have the instantaneous correlation at time 0, and then you have correlations at different temporal lags either side of that. The correlation at 0 temporal lag, the instantaneous correlation between brain regions, is what we think of as a kind of standard resting state functional connectivity that you get in other types of analyses. In DCM, what we actually do is look at all temporal lags. And what that means is that with DCM, as opposed to standard functional connectivity analysis, we're getting a much richer temporal characterization of the relationship in connectivity between different brain regions. And it's going to be the Fourier transform of this cross-correlation function that's going to give us our cross-spectral density. And this is the thing, as I've said, that we're actually going to try and simulate when we're doing our resting state spectral DCM. And what this is showing is, for each of the different brain regions represented by the different lines, the different oscillatory frequencies which these brain regions are oscillating at. And then for each frequency, you have a power associated with that. And what you normally see is a kind of peak for most regions on the left-hand side here, lower frequencies, and then slightly smaller peaks at higher frequencies. This is quite typical of the kind of cross-spectral density that you'll be seeing when you're fitting and simulating your DCM. So that's good. But why is it simpler to do this than to try and estimate the endogenous neuronal fluctuations in the time domain? So in moving from the time to the frequency domain, what you're doing is enabling yourself to no longer have to estimate these time varying neuronal fluctuations. You're parameterizing this and you're having to estimate only a couple of parameters rather than these time varying things, which are very complicated to do. And actually, you just need to estimate their time invariant covariance. And that's what you need to do to understand what's driving the system. This can actually be described by just two parameters, alpha and beta. And these are going to describe the amplitudes and exponents of the spectral density of these neuronal fluctuations, which are driving the system. So just to kind of go over that again, we're moving from the time domain to the frequency domain because we have to estimate these endogenous neuronal fluctuations which are driving our system. By moving from the time to the frequency domain, we're enabling ourselves to only have to estimate two parameters for these endogenous neuronal fluctuations during model inversion. One thing I should say is that this basically constrains the kind of questions you are able to ask a bit as well. So I said earlier on that basically the questions you're going to be asking are going to be to do with group differences. And this is actually constrained kind of mathematically here as well. You can't ask questions about how effective connectivity is changing between regions over the course of the experiment because you're losing that time component. But you can still ask questions about group differences and how effective connectivity differs between groups using this spectral DCM framework. So we've simplified our inversion, our calculation. So that's all well and good, but does spectral DCM actually work well? Can you reliably estimate the underlying connectivity? Yes, you can, which is good. And it turns out that it's not just computationally more efficient, but actually it is more effective and accurate at estimating the true underlying connections than other forms of DCM for resting state fMRI. So this is some work done by Adil Razi, who really pioneered a lot of the spectral DCM work. And this is some data that they have generated here in these plots. So this is some synthetic data which represents the ground truth connections between some imaginary brain regions. And these true connections are shown here by the black bars. And then what they did is they applied two forms of DCM to try and estimate these connections using the same framework which I've been describing. So they used a spectral DCM framework where they moved everything into the frequency domain and estimated these two parameters of endogenous neuronal fluctuations and tried to simulate the cross-spectral densities. And then another form called stochastic DCM, where they actually try and estimate these endogenous neuronal fluctuations in their time-varying form. So this is possible to do, but it's just computationally inefficient. And what you can also hopefully see here is that it's actually producing poorer estimates of the true underlying connectivity. And these estimates are represented by the grey bars here. And you can see that the spectral DCM has estimated and ground truth connections closer together for a lot of these connections than the stochastic DCM does. So it works well and is computationally more efficient, so that's great, but as I said on the last slide, it does assume that the effective connectivity is stationary over time. So the kinds of questions that you're able to answer are constrained to ones about group differences. Although, because you have no task, that's normally the kind of question that you're interested in asking when you're doing resting state DCM anyway. So we have our DCM that we know is computationally nice and efficient and gives us more reliable estimates of the underlying connectivity than other alternative methods. And I just want to spend a bit of time to sort of zoom out and review what the changes we've made are and how this looks in terms of a kind of broader DCM experiment. So this is a figure from Peter's Introduction to DCM paper. I cannot recommend this paper and its partner paper, which looks at the second level group analyses, highly enough. It's an excellent place to start if you want to do some DCM analyses. And what this is showing is an overview of this forward DCM model, which we're specifying is. We've got our experimental stimuli here, driving connections between regions within the neural model. We've then got our hemodynamic model explaining how these changes in circuitry are driving hemodynamic changes and the bold response, and how this is then measurable and the kind of measurement noise that we might expect to be associated. As we've said, there are some key differences. So rather than allowing the circuitry to be driven by experimental stimuli, we allow them to be driven by endogenous neuronal fluctuations, and in our case, the parameterized form of them in the frequency domain, and specifically these two parameters, alpha and beta. These are going to be driving our circuitry, and then rather than trying to simulate time series data, as we've discussed, we're going to be simulating cross-spectral densities. So yeah, this is just the equation again, which I showed you a while ago. The changes in neuronal states over time are based on our hidden neuronal states, our connectivity parameters, and our endogenous neuronal fluctuations. We then have our observation equation, observation function here, G, which shows how the data that we actually are observing is again based on these hidden neuronal states and some hemodynamic parameters based on things like the shape of the bold response and the way that veins and arteries kind of dilate when the brain is active. And yes, that's kind of stuff which has already been modeled and decided upon. So this can actually stay the same in our spectral DCM. We then got this design matrix here, which is going to model things like movement or uninteresting effects that we're not interested in during our scan, but subject level uninteresting effects basically. And then we've got some measurement noise term here at the end. So overall, this is how our first level DCM is going to look for cross-spectral densities. Finally, I just want to talk about model inversion and testing hypotheses. Like I said, if you want to know this in more detail than specifically for the context of resting state fMRI, you can come to the practical session, but I'm going to go over it here as well. So the model inversion is actually going to use the same technique as we would use for conventional DCM. This is something called variational Laplace. And what variational Laplace does is tries to find parameters that can simulate the data in a way that best explains it without straying too far from our prior hypotheses. Formally, this is described by something called the log model evidence. So the probability of having observed some data Y given your model M. In practice, you can't normally directly estimate the log model evidence. So this is approximated by what we call the free energy, or the negative variational free energy. And it's basically finding a trade-off between accuracy and complexity. And this is what variational class is going to do. It's going to be finding parameters that maximize the free energy. And in this way, there's a way of kind of avoiding overfitting, which is very handy. So the parameters that we're actually going to be estimating for our spectral DCM are these ones here. And these are our inhibitory self-connections, our extrinsic connections between regions, and then our amplitude and exponent of endogenous neuronal fluctuations. And here are some prior values here, which are decided upon based on previous validation work. You do have the option to change these if you want, but I don't think you really need to. So that's actually what we're going to be trying to estimate. These specific parameters is what we're going to be estimating when we do the inversion of a first level DCM. And then testing hypotheses. Again, this is going to use identical methods to those used in task-based DCM. And specifically, it's going to use a methodology called parametric empirical Bayes. And what parametric empirical Bayes does is it basically takes our estimated individual level connectivity parameters to the group level and models them as hypothesized group differences. We can then compare between different models that we've built using Bayesian model comparison, or nested models using Bayesian model reduction. By this, I mean we can compare lots of different models that have different connections between regions switched on or switched off. And then we can use that to try and explain what the most important connections are to explain the differences that we're seeing between groups. There's an example of this here at the bottom. This is from a paper which I wrote, which was looking at differences in effective connectivity between Parkinson's disease with and without visual hallucinations. And what you can see here is some Bayesian model comparison showing the optimal model to describe the commonalities and the differences between subjects, and the posterior probabilities associated with these different models. And then here you can see the models which it's pulled out, where it's basically saying, here are the connections which are most important in explaining the commonalities between subjects and the differences between subjects, the differences between your groups. In this case, this was as a result of having visual hallucinations, but it could be anything that applies more directly to the work you want to do. And if you want to know more, you can read my paper or you can ask me about it. So that's it for now. I may see some of you at the practical session, but otherwise, thank you very much for listening. I hope you enjoyed. And just to give you some reading, the kind of papers that I would most recommend are the Introduction to Spectral DCM by Carl and the validation of the Spectral DCM framework by Adil Razi, alongside really strong recommendations for Peter Zeidman's tutorial papers. This is maybe the best place to start. And then up here, if you want to know more about the kind of maths and the models themselves in a bit more detail, you can look at these. And then if you want to see an example application, you can read my paper here. Thank you very much.